네, 안녕하세요. 감사합니다. Today I'm actually going to be presenting my presentation in English. Feel free to please ask me questions later in Korean. I'd be able to uh, try to answer the questions in Korean. However, I will be continuing my presentation in English for today. Okay, so first of all, I'd like to thank the BRIC Biological Research Information Center for providing me with this opportunity to present my work today. And again, my name is Kathy Sung, or my Korean name is Song Ye-won. I'm a postdoctoral fellow at the National Institutes of Health, specifically the National Institute on Deafness and Other Communications Disorder. I also want to share with you that I am an incoming assistant professor at the Department of Biology at University of Iowa. And I'll be starting this position in 2025. So if anybody is interested in this work that I'll be presenting today, feel free to email me at my email. My email will show up in the acknowledgement slide at the end of the presentation. Okay, so I'll just delve into my project. This is the project that was published in Science Advances. I'll be today introducing you on how macrophages, what the role of macrophages is in cisplatin-induced hearing loss. Okay, so I'd like to just briefly introduce you to our ear anatomy. Our ear is comprised of three compartments: the outer, middle, and inner ear. When you hear sound, the sound will enter through the outer ear, the ear canal, that will then vibrate this tympanic membrane, that will then vibrate the ossicles. And then this vibration will enter the cochlea, which is the hearing organ. And the cochlea is where this mechanical sound energy gets converted to an electrical sound energy that then gets transferred through the spiral ganglion neurons that will reach the brain for sound recognition. Within the cochlea, there's an organ called the organ of Corti, and within this organ of Corti, you see these sensory hair cells, which are very specialized hair cells, and there are two types of hair cells, the outer and inner hair cells. The outer hair cell and inner hair cells are both critical in hearing, and if you have a loss of either of these hair cells, then you do have significant hearing loss. And so I'd like to just briefly show you a 3D structure of the ear. So this is a 3D orthographic rendering of a human cochlea. And you can see the hair cells that are lined along this basilar membrane that supports the hair cells. And you can see it spiraling along the structure called the medullus. And what this allows the cochlea to have is what's called a tonotopic organization. And so what this is that in the apical region of the cochlea, so the hair cells that are in the apical region will detect sound that are at low frequencies. And then as it moves along this basilar membrane, the hair cells will detect increasing sound frequencies, with the hair cells that are located in the basal region detecting the highest sound frequency. Now, this, you can think about it through a piano keyboard. The apical region will detect sound that is at the most left side of the keyboard, so the low sound frequency, and the hair cells that are in the basal region will detect the sound that's in the high on the right side of the keyboard, so the high sound frequencies. Now, when you think about hearing and the frequency range that we can hear, you can imagine that depending on the different species and where they live and their environment that they live in, they might have very, very different ranges of frequencies. And so for us humans, we can hear between 20 hertz and 20,000 hertz. And as opposed to what we can hear, mice, which are frequently used for a lot of our experiments, hear a much higher sound at 1,000 hertz to 90,000 hertz. And so you can see that there is this very big difference in terms of the frequency range mice and human hear. However, when you look at their cochlea, the organization and the structure of the cochlea are very similar, and also the cells that make up the cochlea are very similar between these two. And so when you look at the mouse cochlea, this is a dissected mouse cochlea and stained for myosin 7A, which is a marker for sensory hair cells. And you can see these sensory hair cells that are spiraling along, with this part being the apex and the other side being the base. And you can see that this is frequency mapped. And so this region of the hair cells used to detect 4 kilohertz sound, and this part of the hair cell used to detect 64 kilohertz sound. So here, throughout my presentation, I'm going to be showing you data as well as images of different regions that correspond to the frequencies here indicated here from 8 to 63 kilohertz. And so why do we study the ear? The main reason is because hearing loss is the most common sensory disorder, and approximately 430 million individuals worldwide are affected by this hearing loss, and about $750 million annually are spent to treat hearing loss, and also by 2050, over 700 million people will and is estimated to have this disabling hearing loss. And so it's really important for us to continue to study hearing loss and continue to identify causes and underlying mechanism of which hearing loss occurs, and so that we can also develop therapeutic drugs to prevent hearing loss. So what are the causes of hearing loss? There are a number of different causes of hearing loss, but the most common causes of hearing loss are the first three here. The genetic mutation, so a mutation in a deafness gene could cause hearing loss. There are many deafness genes that has been identified, as well as many mutations within those deafness genes. And then you can also get hearing loss if you're exposed to loud noise. And also for humans and other mammals, as we age, also lose hearing as well. And then there are other causes such as viral infection or bacterial infection in the ear, and then physical head trauma, which, say, for example, if you have a concussion and you also have some sort of trauma in the ear as a result of that, then you can also have hearing loss as well. And then finally, there is ototoxic medications. So two of the many actually are the aminoglycoside antibiotics. And so some of these you may have heard: amikacin, kanamycin, neomycin, streptomycin, gentamicin, and tobramycin. These aminoglycoside antibiotics can cause significant loss of hair cells, and as a result can cause damage or loss of hearing. And then also a drug called cisplatin. And cisplatin is a potent anti-cancer drug, but also comes with significant ototoxicity. And so today I'm going to be focusing on this cisplatin-induced ototoxicity. So cisplatin is among the most widely used and very effective anti-cancer drug that treats a variety of different types of cancers, including head and neck cancer. And if you look at the structure of cisplatin, you see that cisplatin has an ammonia on the side. And then originally cisplatin has chloride on the other side, but when it enters the cells, it goes through a process called equation, and it exchanges the chloride to water molecules. And these water molecules and also the cisplatin, the center of cisplatin is made up of platinum. Now, these water molecules are actually very important because this exchange between the water molecule and chloride makes the cisplatin become much more reactive with DNA, and therefore it actually binds and forms the cisplatin DNA adducts. And because it makes these adducts, you can imagine that it interferes with transcription and therefore translation, as well as protein synthesis, which are the downstream processes. And so when cells undergo stress because of these DNA adducts, they can undergo DNA damage response or other stress response, and then they can also lead to cellular death through apoptosis. Okay, so this can actually be very helpful in treating cancers, especially cancer that are actively proliferating. But unfortunately, cisplatin does cause very significant side effects. But the two main side effects that are well known and associated with cisplatin is the ototoxicity and nephrotoxicity. And so today I'm going to be mainly focusing on ototoxicity. Now, among patients that receive cisplatin, about 50 to 60% of them will develop the cisplatin-induced hearing loss in forms of sensorineural and bilateral hearing loss. Now, sensorineural hearing loss means that you have hearing loss because there's a damage in the cochlea that I've mentioned to you earlier. And also, cisplatin causes hearing loss with a known mechanism due to causing death of these hair cells. So these hair cells, and specifically these outer hair cells. And because mature mammalian cochlear hair cells can't regenerate, they don't have the capacity to regenerate. Hearing loss in these patients are mostly permanent and almost permanent. Now, another property of cisplatin is that cisplatin initially causes hearing loss at the high frequency region, so it causes damage to the hair cells that are located in the basal region. But with continued treatment and higher cumulative dose of cisplatin, it will then further progress to the lower frequency region that can also affect our normal hearing, normal conversational range. Now, I also wanted to indicate that cisplatin can cause nephrotoxicity, and more than 30% of patients treated with cisplatin will develop the cisplatin-induced kidney injury. And one of the studies that came out about 2 years ago was really interesting to me because it indicated that depleting the kidney-resident macrophages ameliorated cisplatin-induced kidney injury. And so that essentially means that macrophages have somehow promoted or somehow played a pathogenic role in the development of kidney injury. And so this was really interesting to me, and I further wanted to assess whether macrophages were also doing something very similar in the development of cisplatin-induced hearing loss. And so I guess the first thing that we should address is, are there macrophages in the cochlea? And so what we do know about macrophages in the cochlea is that there's a lot of macrophages in the cochlea. More than 95% of the immune cells in the cochlea are macrophages, making them the major immune cells in the cochlea. And so this is a cochlear section of a mouse, a cochlear section of a mouse. It's a transgenic mouse where CX3CR1 GFP positive or CX3CR1 positive macrophages were labeled for GFP or green fluorescence protein. And so you can see a lot of these GFP labeled macrophages that are all scattered throughout the cochlea. And so the macrophage function in the cochlea are very similar to other tissue-resident macrophages found in other organs. They promote and maintain tissue homeostasis. They are usually the first responder to local damage. They also produce inflammatory response. They are the professional phagocytes, and they also play a role in tissue repair responses. Now, I also wanted to bring you to this region right here. This is one cochlear turn and show it to you through this schematic diagram. And the macrophages here are shown in magenta. And I wanted to specifically draw your attention to this organ here called the stria vascularis. The stria vascularis, also located here, has macrophages as well. And these macrophages are a little bit more unique. And they're called the perivascular macrophages. The stria vascularis has a very dense capillary network. That's where what's called the blood labyrinth barrier is located. Now, blood labyrinth barrier is very similar and analogous to what we know about the blood-brain barrier in the brain. And so again, the stria vascularis is highly vascularized. And you can see that these CX3CR1 positive GFP macrophages are also very closely associated with these blood vessels that are labeled with CD31. And so these macrophages are called the perivascular macrophages. And again, they're very closely associated with these blood vessels. And there's been a couple of papers and studies that came out indicating that these perivascular macrophages can also regulate this vascular permeability. And so these perivascular macrophages can increase the vascular permeability, which it means that there's a lot more exchange between the blood and the cochlear fluids, but sometimes it could also maintain the integrity, minimizing the exchange between the blood and the cochlear fluids. And so with that being said, we know that there's a lot of macrophages in the cochlea. And so the main objective of my study was then to examine the roles macrophages play in cisplatin-induced ototoxicity and to identify the mechanisms underlying their activity in response to cisplatin. And so the way how I approached this was using a pharmacological approach. So when you look at a macrophage, macrophages express a receptor called CSF1R, or colony stimulating factor 1. This is a receptor that has a ligand, CSF1, or colony stimulating factor 1, or IL-34. When it binds to this receptor, it'll activate the downstream signaling pathway that will result in microglia, so the macrophages in the brain, microglia as well as other macrophages, their maintenance and survival. Now, this drug called PLX3397 or pexidartinib is a CSF1R antagonist and an inhibitor. And so when this drug binds to the CSF1R, it'll inhibit the downstream signaling pathway, further resulting in microglia and macrophage ablation. And this has been very well documented in the brain. The PLX3397 does very effectively ablate microglia. Now, another aspect about using this drug and a benefit of using this drug is that it's already approved by the FDA to treat tenosynovial giant cell tumor. And so, let's say, for example, if PLX3397 had prevented against cisplatin-induced hearing loss, then it makes it easier for us to then repurpose this drug to treat cancer patients that are receiving cisplatin to prevent hearing loss. And so we wanted to then specifically address the question using this drug: do macrophages contribute to hearing loss and mediate hair cell death or survival in response to cisplatin treatment in vivo? And so to address this research question, we utilized a cisplatin administration protocol that was previously developed in our lab. We first do the hearing test for these animals at the baseline, followed by a 4-day cisplatin treatment and a 10-day recovery period, and this encompasses cycle 1. We performed this in 3 cycles, and at the end point, we again performed the hearing test. And then after the hearing test, we harvest the cochlea for any downstream experiments. Now, to address the role of macrophages, I've mentioned to you that we would use the PLX3397. So here we utilize this drug in chow form to administer into these animals. We administer them 7 days prior to cisplatin administration to pre-ablate the macrophages, followed by PLX3397 oral gavage daily during the cisplatin administration. And the 4 groups that we utilized for this study was saline vehicle, saline PLX to ablate the macrophages, cisplatin vehicle, and cisplatin PLX 3397, again to ablate the macrophages. And so we were first interested in wanting to understand whether PLX3397 ablated cochlear macrophages. We know that PLX3397 ablates microglia in the brain, but but it wasn't very much known whether PLX3397 ablated cochlear macrophages. So here, after treating the animals, we took the cochlea and sectioned them. And here what you're seeing is a cross-section of the cochlea that's stained for DAPI, the nuclear stain, as well as KIR 4.1, which is a potassium channel. However, here for the purpose of this image, KIR 4.1 was used as a landscape marker to indicate the spiral ganglion neuron regions, the organ of Corti regions, as well as the stria vascularis region. Now, I'd like to draw your attention to this medullus, the medulla region, as this is where we see a lot of macrophages. So here you can see the macrophages in green, the CXCR1, CX3CR1 GFP positive macrophages. And when you quantify them in the whole cochlear section, you can see that there are a number of macrophages present in saline and cisplatin treated animals, even though there is about 20-25% reduction in the number of macrophages. However, in animals that were treated with PLX3397, there were more than 95% reduction or ablation of macrophages. We also checked whether PLX3397 ablated perivascular macrophages in the stria vascularis. And so here again, we've performed and stained the stria vascularis with CD31. So here you're seeing the blood vessels, as well as visualized CX3CR1 GFP positive macrophages. And we quantified these perivascular macrophages and found something very similar where there were a number of macrophages present in both saline and cisplatin treated animals. However, there was a slight reduction in the number in cisplatin treated animals. Now, again, in animals that were treated with PLX3397, there were more than 90% reduction in the number of macrophages. And so this indicates that PLX3397 effectively ablated both cochlear macrophages as well as perivascular macrophages. And so then we delved into our very important question, which is, does macrophage ablation affect cisplatin-induced hearing loss? And so here we performed the hearing test for animals. This is called the auditory brainstem response. Here we provide sound that are at different frequencies from 8 to 40 kilohertz at different sound pressure level, and we determine the threshold of which these animals can hear. And so the threshold that were obtained here at the baseline of the hearing test and at the endpoint of the hearing test, the difference between these two thresholds are recorded here as threshold shift. And what would be important to know is that the zero would indicate no hearing loss, and 60 would indicate severe hearing loss. And so what we've identified is that in saline treated animals, when they were given this PLX3397, there was no threshold shift, indicating that these animals did not have hearing loss. Now, animals that received cisplatin, you can see that there were significant threshold shift, suggesting that these animals have developed hearing loss. And then animals that received cisplatin and PLX3397, having ablated the macrophages, had threshold shifts that was comparable to those that were receiving saline. And so I just wanted to just bold this and just emphasize that both animals in the red as well as the green have received cisplatin, but in the presence of macrophages and in the absence of macrophages. And so this reduced ABR threshold shift in the absence of macrophages indicate that macrophages contribute to hearing loss in cisplatin-treated mice. And so we next wanted to identify whether this was associated with the preservation of hair cells. And so I've mentioned to you that cisplatin does cause significant loss of hair cells. And so here we've assessed the number of hair cells in our 4 groups. So in the dissected cochlea, we've stained the cochlea with myosin 7A, again staining for the hair cells. You can see the 3 rows of outer hair cells and a single row of inner hair cells. And in both saline-treated animals that were either vehicle or PLX3397 treated, the number of outer and inner hair cells were similar. In animals that were treated with cisplatin, you can see that the number of outer hair cells were significantly reduced, specifically with it being more severe at the higher frequency regions. And so you can see that you can see this loss of outer hair cells in animals that received cisplatin. And then animals that received both cisplatin and PLX3397 have shown a significantly greater number of outer hair cells compared to those that received cisplatin and vehicle. The inner hair cells were not affected in any of these 4 groups. And so this increased outer hair cell survival in the absence of macrophages indicate that macrophages contribute to outer hair cell death in cisplatin-treated mice. And so this was really striking to us. We were very interested in wanting to understand what the mechanism is that where the macrophages confer this significant and complete protection. And so we further assessed and looked into how cisplatin gets into the cochlea and whether cisplatin entry into the cochlea was affected by macrophages. And so if there's less cisplatin coming into the cochlea, then there could be less ototoxic effects within the cochlea. And so what we know about cisplatin entry into the cochlea is that it comes through the blood labyrinth barrier that I've mentioned to you briefly earlier. I've mentioned that the stria vascularis is very highly vascularized, and these within the stria vascularis is the structure called the blood labyrinth barrier, which again is analogous to the blood-brain barrier. And the blood labyrinth barrier controls the entry of solutes that are within the blood into the cochlea. And so when you receive a systemic administration of cisplatin, the major route of cisplatin entry is through this blood labyrinth barrier into the stria vascularis that will then enter the scala media, where there's a fluid called endolymph, and then the cisplatin will be exposed to hair cells as well as other cells within the cochlea, and it'll then enter into the hair cells across their apical membrane, and cells that are more susceptible to cisplatin will then undergo cell death, especially these outer hair cells. And so just to kind of briefly explain to you the structure and the anatomy of the blood labyrinth barrier, it's comprised of endothelial cells that are tightly connected by tight junctions, and you have pericytes that surround the endothelial cells, as well as these perivascular macrophages. And so I just wanted to remind you that these perivascular macrophages surround the blood vessels and very closely associated with these blood vessels. And there has been studies that indicate that these perivascular macrophages can affect the permeability of the blood labyrinth barrier. And so really the hypothesis and the question was that could macrophages would macrophages regulate this BLB permeability and affect the cisplatin entry into the cochlea? And so the research question that stemmed from this hypothesis is that does macrophage ablation via PLX3397 affect cisplatin accumulation in the cochlea? And so we addressed this research question by using a inductively coupled plasma mass spectrometry to measure platinum content. So here, again, is a cisplatin molecule with platinum in the middle. And the ICP-MS can detect the platinum content in tissues. And so here we've assessed the level of platinum cisplatinum-inspire ganglion neurons, the organ of Corti, the stria vascularis, as well as the spiral ligament. Now, it's really important to note that normally in our cells, we don't have platinum. And so here in animals that were treated with saline, you can see that there was no platinum in these tissues. Now, in animals that were treated with cisplatin, there was significant increase in the platinum level. And what was interesting was that in animals that were treated with both cisplatin and PLX3397, and some of these organs and tissues, you can see that there were significant reduction in the level of platinum, and the stria vascularis had shown a significant reduction in the level of platinum, suggesting that, and actually the stria vascularis being the most important because that is what we know in which cisplatin gets into the cochlea. And so this reduced platinum level in the absence of macrophages indicate that macrophages may contribute to cisplatin entry into the cochlea. And we think that it's through regulating the blood labyrinth barrier permeability. And so just to briefly summarize, the macrophage ablation, so the group that had received both cisplatin and PLX3397 showed complete protection against cisplatin-induced hearing loss, showed greater outer hair cell survival, and also resulted in decreased cisplatin accumulation in the cochlea. And so here we conclude that macrophage ablation protected against cisplatin-induced hearing loss and outer hair cell death, in part by limiting cisplatin accumulation in the cochlea. And so the mechanism that we think is happening is that the macrophages here during cisplatin treatment somehow increases the permeability of the blood labyrinth barrier, allowing more cisplatin to enter the stria vascularis, followed by its entry into the scala media, exposing more cisplatin to the cells within the cochlea, including the hair cells. And when macrophages are absent, the integrity of the blood vessels are maintained. So there's minimal exchange between the blood and the cochlear fluids, and therefore there's less cisplatin that enters the cochlea, and therefore many of the cells within the cochlea are protected because there's less cisplatin overall that have entered the cochlea. And so there are many different future directions for this project. So I've shown you today that PLX3397 completely protects against cisplatin-induced hearing loss. And so we can think about this in two different directions. One being a little bit more clinical in that we could use PLX3397 to repurpose this drug to be able to treat cancer patients that are receiving cisplatin. But before we really do that, one of the more important questions that we need to address is whether PLX3397 affect the anti-cancer efficacy of cisplatin. And in order for us to do that, we need to develop a model where animals have both cisplatin-induced hearing loss and tumor bearing. And so we're currently trying to develop a tumor-bearing mouse model that also incorporates cisplatin-induced hearing loss. And using that mouse model, we hope to also investigate the contribution of macrophages in cisplatin ototoxicity, something very similar to what we've done in our current mouse model that does not have tumor. From a basic science perspective, we'd like to investigate the cellular and molecular mechanism in which blood labyrinth barrier permeability in the cochlea. And so whether it's the soluble factor that's affecting the permeability or whether there's other mechanisms, it would be a very interesting aspect for us to understand. And in addition, we'd like to identify therapeutic targets that protect against cisplatin-induced ototoxicity, in the presence of macrophages. And so the reason why this is very important is because macrophages are also very important for protecting us against bacterial infection or other insults and diseases. And so potentially, if we could just prevent cisplatin-induced ototoxicity while preserving and while in the presence of macrophages, that would be more of the ideal situation. So with that, I'd like to acknowledge everyone who helped throughout this project, especially my mentors, Lisa Cunningham and Mark Worshall, and then many of the individuals who have also helped throughout the project. I also would like to acknowledge Nicole Schmidt, who's been helping out with the tumor-bearing mouse model, as well as Clint Allen and Yvette Robbins, who again also helped out with the tumor-bearing mouse model that we're currently working on. I'd also like to acknowledge our collaborators at the NIDDK Renal Diagnostic and Therapeutics Unit. Today I haven't really introduced you to our kidney story, but we also have seen that PLX3397 significantly and almost completely protects against cisplatin-induced kidney injury. And then I'd also like to acknowledge my husband, Alex Kleinpeter, for his support and also my funding source that includes the funding from NIDCD as well as my K99 and then my email address. And I'd also like to thank everybody for joining today. I'd like to end by introducing you to this YouTube video. This YouTube video is actually a fun video to try out. So it actually starts it'll produce sound that starts from low sound frequency to high sound frequency, and you can determine how old your ear is essentially by determining when you start to not hear the sound that it's producing. And so this is just the QR code for you to be able to get to that YouTube video. And with that, I'd like to thank everybody again for your attention, and I'd be more than happy to take any questions. Thank you very much. 감사합니다.